Alright, so in this video, I'm just going to be talking about the in-class activity about translation that you guys did. So yeah, let's get started with it. So first of all, let's look at our given. We are given a wild type coding strand and also a cystic fibrosis coding strand. Now, just one thing I want to tell you guys about wild types. When you see the word wild type or the word wild type, it means that it is the normal sequence, it is the naturally occurring sequence, or like the healthy, like quote unquote healthy sequence. And then the other one is obviously the mutated sequence because there's something wrong or something that is missing from the wild type. So yeah. So in this case, this is the wild type, this is the naturally occurring one, and then this is the mutated type of strand. As you can see in here, this one is a lot longer, this one is a lot shorter. Alright, for number one, the first thing it asks us to do is to transcribe the wild type and the cystic fibrosis DNA into an mRNA. Now, look at our given. We are given the coding strand over here. And I would suggest also to watch the other videos that we made because we explained what coding strand, template strands, and mRNA strands are. But in this video, when we are given the coding strand and when we are asked to give the mRNA, all you need to do is literally just copy everything else and then change the T with a U. Alright, let me just write everything out first just to make it easier. I'm just gonna rewrite it. And then I'm gonna do it over here too. And by the way, when you see like it's like small letter format, like don't care about that. It literally just wants it's just the way to show that there's a mutated sequence on it. So yeah. Alright. Now I copy this. Since we are given the coding strand, when I'm writing the mRNA strand, all I have to do is copy it again, except change the T into U. So what I meant by this, it's gonna be the same thing. It will also be 5'. And instead of ATC, I'm gonna write AUC because this is the coding strand, and coding strand will look similar to the mRNA strand. So I'm gonna rewrite everything: AUC, AUC, and then UUU, GGU, and then GUU. Same thing also, it will be on the 3' end. And then I can also do the same thing on the other one. This will be our mRNA strand for this one. Alright, yeah. Now, next thing that it's asking for is to translate it. What we did first, remember when we're making an mRNA, we're transcribing it. Transcribing is literally just transcription because we're making an RNA strand from the DNA sequence. So once we have the mRNA over here, you know what, I'm gonna write it over here. mRNA. Same thing over here, this is also the mRNA. Now we can translate the mRNA into the protein sequences using 3-letter codes. Now, I would also suggest watching Kenzie's video. She has a very fun mnemonic type of way or type of memorizing the amino acids. Maybe that will help you. So let's try. For this one, we also need to use the codon chart. Now the codon chart, it will be given to you in the exam. Don't worry about memorizing that one. So look at your codon chart, and then a codon, when you're reading codons, it has to go 3 by 3. What I mean by 3 by 3 is that you have to read 3 nucleotides each time. And this is what a codon is. A codon is reading it 3 nucleotides each time. And you have to read it from 5' to 3 prime. Always 5 prime to 3 prime when you're reading a strand, especially for translation. Always read it from 5 prime to 3 prime. If you wrote it like, let's say you wrote something like this way instead, 3 prime to 5 prime, you gotta read that the opposite way, because you always have to read it 5 prime to 3 prime. Alright, so let's start with the translation. AUC is isoleucine. UUU is phenylalanine. GGU is glycine. And then GUU is valine. I'll do the same thing for the other one. AUC is another isoleucine. AUU is also isoleucine again. We have glycine and then valine. Now, don't ignore this yet because we're not done with it. Number 2 is asking us to add the appropriate labels to both the mRNA and the protein sequences. The first part is already done because we already labeled our mRNA. We already added the 3 prime and the 5 prime. Same way over here, we have the 5 prime and 3 prime here. That is the right labeling of a strand. When we're dealing with DNA and mRNA, we always have to make sure that we write the 5 prime and the 3 prime polarities. And then for protein sequences or polypeptide sequences, we don't use 5 prime to 3 prime. Instead, we use something called N-terminal or C-terminal. Now, there are different ways to write the N-terminal and C-terminal. Alright, so don't ignore these letters over here. Sometimes the N-terminal may be written this way, like literally just the N-terminal itself. It could also be written as H3N plus or H2N. And then for the C-terminal, same way, it can either be written literally as C-terminal itself. It could also be written as COO minus or COOH. For those of you that are more chemically inclined, I would suggest just using either one of these. But for the sake of this class, I would suggest sticking with just one format. For me, normally what I use for the N-terminal is that I use H3N plus, and then I use COO minus for the C-terminal. But you can use either one of them. There's no difference between using either one of them. Let's go back to the activity. So for this one, normally the 5 prime end over here will correspond to the N-terminal. So we can just assume that this one is the N-terminal. Remember, this is what I assumed it to be. This is the format that I'm normally accustomed to using. You can also use H2N or N-terminal itself. And then for 3 prime, it corresponds normally with the C-terminal. And this is the format that I use, COO minus. Same thing for the mutated patient. I use H3N plus and then COO minus for the C-terminal. Alright, number 3. It literally just says rewrite this sequence over here, but instead of using the ILE for the name of the amino acid, we're gonna use the 1-letter code. So what is the 1-letter code of isoleucine? Is letter I. And then for glycine is G. And then valine is V. Alright, we're not done here yet. We need the N-terminals and C-terminals again. Always, when we're dealing with protein sequences, always put the N-terminal and the C-terminal. So we write our N-terminals and C-terminals over here. This is called C-terminal because there's a C on it, and then N-terminal because there's an N on it. Like, if you see it this way, the N stands for nitrogen, the C stands for carbon. So yeah. Alright, what type of mutation did we find in this patient? Let's go back to our given. Now, let's look at this. We have ATC. We also have ATC on the bottom. We have ATC at the second one. We don't have it here. TTT, we also don't have it here. And then GGT, we have it. GTT, we have it. So what we're missing is this C and these two T's over here. So that means both these three nucleotides over here got deleted. And then that type of mutation is called deletion. Literally, like, because it got deleted, that's why it's called deletion. There's different types of mutation as well. I will make a separate video about that. But for this activity, these nucleotides over here were deleted. And then we resulted in this sequence. Alright, number 5. It says underline the amino acids with non-polar characteristics and then circle the ones with the polar. So if we're looking at this protein sequence, everything, isoleucine, phenylalanine, glycine, and valine, all of them are non-polar. I'm sorry. All of them are non-polar. Because if you look at the amino acid structure or amino acid chart, all of them are non-polar, and then we don't have any polars here, so we don't have to circle anything. Alright, last one. The last one is we have to draw the actual structure of the amino acid. But I just want to remind you guys, for the exam, you guys won't have to memorize the actual structure. You're only required to memorize the 3 letter codes and then the 1 letter code of the amino acid. But for this one, it's just asking us to draw it and then show the peptide bond. I also made a video about it. I would suggest watching it. But in this video, I'm just gonna go over how peptide bonds are formed. I will try to go over it pretty quick. All right. Amino acids. Amino acids are mostly composed, like it has this general molecular formula. It has this amine group, which is H3N plus or H2N. I'm just going to put H2N. But in this case, I'm just going to use H3N plus. And then it also has a carboxyl group. It could be COO minus or COOH. Now let's put this together. An amino acid will have an amine group, a central carbon, and also a carboxyl group. It also has a hydrogen group over here, and then a specific R group. Now this R group is what literally makes, or like what gives the name of the amino acid, because it's very specific for each amino acid. Alright, now how does the peptide bonds form? Let's say we're gonna connect this with another peptide bond, right? So when I draw an H3N plus out, it will look like this. H, H, H. Same, then I'm gonna copy the same amino acid formula. We have the central carbon, a carboxyl group over here, an R group, and also an H group. Now, what happens here when we form a peptide bond? Two of the H's, whichever two of the H's that are connected with the nitrogen over here, and then the O minus over here is gonna form a molecule called water or H2O. And then once those molecules formed, you're literally just gonna connect this carbon over here to this nitrogen over here. So in the end, you will have this final structure of like this. So as you can see, we lost the O minus on here. We also lost two of the H's over here. And then we had a bond that formed between the C and the N, which is this one. Now, literally this bond over here, this is your peptide bond. So, just to summarize it again, every time we form a peptide bond, we lose water with it. Alright, let's go back to the activity. For activity, it says draw it out. It's including the actual specific R group. So I drew it out over here. This is our mutated peptide sequence. It's isoleucine, isoleucine, glycine, and valine. So these over here, these are the R groups from what I drew earlier, like the general amino acid formula. These are your specific R groups. This corresponds with isoleucine, isoleucine, glycine, and valine. Now, I will try to make this as simple as possible because I don't want to confuse you guys. This N-terminal over here corresponds to this H3N plus over here that is connected to the first amino acid. That's why we have the N-terminal. Same logic can be applied for the C-terminal. The C-terminal corresponds to the carboxyl group found in the last amino acid of the peptide sequence. So that's why we have the N-terminal in the start and the C-terminal at the end of a peptide sequence. Now, when we're trying to figure out which one is the peptide bond, remember, general formula is that we have an amine group, a central carbon, and then another carboxyl group. But in this case, it's already connected, so that means this is our first peptide bond. Same logic again, we have the amine group over here, central carbon, carboxyl group, and then peptide bond. Same again, peptide bond. So yeah, that is what literally number 6 is asking for. Draw the whole structure and just circle the peptide bond. And this is it. So yeah, that is the video for this. That is it for this video, and I would suggest watching other videos as well to make sure that you're understanding every language we've used here. We're also gonna be uploading more videos about translation itself. So if you need more help, email us and we will try to answer to the best we can. Alright.